introducing Catherine Faivre-Sarralh, who is the director of research at the Neuroscience Institute in Marseille, North Marseille. Catherine is an old friend and colleague, and we have a lot of, parallel pathways in our research, as she started working with neuronal adhesion molecules and neuronal development, and then I think focused more in her own lab on axoglial interactions with these particular molecules. And so she will, as we did pretty much. So you will hear more about her recent research in the center. Okay, Catherine, thanks for coming.
Thank you, Donna. Yes, when I came first in our field, it was more than 10 years ago. Donna said more than 15 years ago. But at that time, I was mainly working on axonal growth and guidance. And now I switched to the field of myelination. So I will talk in the first part of my talk. I will present you some recent data about cell adhesion molecule, which are implicated as immune target in some peripheral inflammatory neuropathies. And in the second part of my talk, I will show you unpublished data about the formation of paranodal junction and juxtaparanodal junction using myelinating cell culture. So let's start with the classical introduction for the people working on the node of Faivre. So this is the ultrastructure of a node of Faivre, and here is the schematic drawing. Here is a nodal gap, which is highly enriched with sodium channels, voltage gate sodium channels at the basis of a rapid saltatory conduction. And here is, on this electron microscopy, I image the nodal gap, which is bordered by the paranodal loops. This is the myelin terminal loops, which here are anchored to the axonal membrane via a very specific type of cell-cell junctions, which are called the septate-like junctions, because you can see here their feature with the transmembrane particles, which are regularly aligned between the glial and the axonal membrane. So here you can see on these immunofluorescence staining images the sharp segregation between the three domains, the three axonal domains of the node of Faivre. So in the center, in green, this is the concentration of the voltage gate sodium channels. In blue, this is a staining for Caspr, which is a cell adhesion molecules, which is implicated in the formation of these septate-like junctions here. And in red, this is the juxtaparanodal region, which is highly enriched in voltage gate potassium channels. And these channels, these potassium channels, ensure the resting potential at the level of the internodal region. So here, the juxtaparanodal region are under the compact myelin here. So we are interested in understanding the mechanism of axoglial interaction at this level, and how these cell adhesion molecules and the ion channels can be segregated all along the axon. So the septate-like junctions were first identified in the fly. So this is a special slide for Mara Strigini. And so here is a comparison between the junction, the cell junction in epithelial cells in vertebrates and in invertebrates. And in vertebrates, the tight junctions are acting as paracellular barriers, and they are formed by these strands of kissing points here, which are made by the oligomerization of a protein, which is a 4-pass transmembrane molecule, which is a claudin. Well, in the fly, there are different types of cell junctions. Well, the adherence junctions are well conserved during evolution. But there is no tight junction in invertebrates, but septate junctions, which are the functional equivalent of the tight junctions. So the septate junctions here are made of these transmembrane particles, and you can see here the strands of septate, which are aligned here. So this is a very different ultrastructure of these junctions, but they are playing quite the same role, providing a paracellular barrier. So the molecular components of septic junctions were identified nearly at the same time in the paranodal junction and in the fly septate junctions. The neurexin-4 is a cell adhesion molecule which was first identified by the group of IMBB in the fly septate junctions, and then Caspr paranodine was identified at the level of paranodal junctions, both by the group of Antoine Giraud and by the group of Oripelez. Next, two other cell adhesion molecules were identified to be implicated in the formation of septic junctions. They are Contactin and neuropathies 155, which are members of the immunoglobulin superfamily. And at that time, we identified also in the fly the homologue of Contactin, which is Drosophila Contactin, and neoglia, which is the homologue of neuropathy. And these three molecules form a ternary complex and are required for the formation of the septic junctions. So, what is the role of the paranodal junctions at the node of Frankie invertebrates? Well, so let me start with a classical interaction about the different molecular complexes that are formed at the node, paranode, and juxtaparanode. So we have already seen that the paranodal junctions are made of Caspr, Contactin, expressed at the level of axon, and facing neuropathin 155, which is expressed by the glia loops. Here, at the level of the cytoskeleton, Caspr interact with a protein which is a scaffolding protein of a firm family, which is 4.1b, and this protein interacts with underlying cytoskeleton and with the component IMBB and spectrin beta 2. At the nodal region, the voltage gate sodium channel are enriched at this site because they are clustered with the scaffolding molecule ankyrin G, which interact also with spectrin beta 4. And ankyrin G binds the sodium channel, but also binds a cell adhesion molecule, which is neuropathin 186. So it's different splicing isoform, which are expressed by the axon and by the myelinating glial cells. And these cell adhesion molecules are clustered at the nodal region because they interact with the extracellular matrix component, mainly the gliomedin, which is identified by Oripelez in the peripheral nervous system. So gliomedin here is secreted by the Schwann cell microvilli and induces the clustering of this complex. Well, on the other side, at the juxtaparanodal region, so as you may know, because Tom worked a lot about the juxtaparanodal junctions, the potassium channels, the tetrameric potassium channel Kv1.1, Kv1.2 are clustered at that side because they are interacting via unknown scale. For at the moment, it is not clear what are the scaffolding molecules which are implicated here in the formation of this complex, but the complex is formed because of the interaction with cell adhesion molecule Caspr2 and Tag1. So Tag1 is also called in the human Contactin 2, and it's really close to Contactin. It's the same family, and Caspr2 is homologous to Caspr. So this is quite a similar complex, ternary complex, which is formed at juxtaparanode and paranode. So what is the function of a paranodal junction? It's to act as a fence separating these two domains enriched in sodium and potassium channels. As you can see here, in the knockout mice for either Contactin or Caspr, or mice deficient for neuropathin 155, there is an alteration of a paranodal junction, and as you can see, this is the wild type here, and here are the picture of a knockout mice. You can see the disappearance of a transverse band between the myelin loops and the axons. And the consequence is that, well, the region, the nodal region is enlarged, And the most striking phenotype is that the potassium channels are now have moved to the paranodal region. So here is a wild type situation where the potassium channels here are well separated from the sodium channels, and here in the knockout mice you can see the potassium channels which are really in the vicinity of a sodium channel. So the physiological consequence is that there is a decrease in the velocity of action potential propagation. So it means that the safety junctions act as a fence and have a very critical role in regulating axonal conduction. So now I will talk a little bit about the formation of this complex and the crucial role which is played by the N-glycosylation in this process. So Caspr associates with Contactin, and both molecules are expressed at the level of the axon. And we showed, it was a long time ago, that the complex associates in cis early during the exocytosis pathway in the endoplasmic reticulum. In fact, when Caspr is expressed alone, is transfect alone in here it's in neuroblastoma into A cells, you can see it is blocked within the endoplasmic reticulum. And when Contactin is co-expressed with Caspr, you can see that Caspr is nicely expressed at the cell membrane. So we showed that short motif in the extracellular domain of Caspr, which consists of several repeats of a PGY amino acid, act as retention motif. And when Contactin associates with Caspr, it adopts a transport permissive conformation, and the complex is exported at the cell surface. And next, what we showed is that the complex is exported at the cell surface bearing a specific type of N-glycans, which are mannose-rich N-glycan. Well, we perform cell surface biotinylation and then immunoprecipitation, and we show that the complex of Caspr and Contactin is sensitive to NOH, endoglycanase H, meaning that they bear only mannose-rich N-glycans, which are typical glycans of endoplasmic reticulum. So when Contactin is expressed alone, it is processed through the Golgi apparatus, and it bears complex N-glycans, including sialic acid and galactose, etc. And as you can see here, it does not bind the glial ligand neophasin 155. But if we use the mutant cells, the LEC1 mutant cells, which are mutated for the N-acetylglucosamine transferase, and which induce the expression of glycoprotein with only 5 mannose at the cell surface, you can see that this, in these cells, Contactin strongly binds neophasin 155, meaning that the trafficking and the glycosylation of Contactin and Caspr is really crucial for the formation, for the interaction with the glial partner, and for the formation of the paranodal junction. So next, we map the N-glycosylation sites of Contactin, which are implicated in the binding of neophasin, and it was the work of Marie Navas, who was postdoc in the lab, and she mutated all the N-glycosylation sites of Contactin, and she observed. I don't know if you see what. Yes, I don't know if you see where the binding. Well, it's in red. The binding of neophasin is in red, and Caspr and Contactin GFP is expressed in the LEC1 mutant cells. And you can see here that mutation of this cluster of N-glycans in the IG5 domains completely prevented the binding of neophasin 155, meaning that this cluster is implicated in neophasin binding. But this mutation did not induce a stronger structural perturbation of the molecule, because as you can see here, the mutant Contactin is able to interact with Caspr and to induce the cell surface expression of Caspr. Well, we have this data, and at that time in the lab, we were interested in the implication of cell adhesion molecule in some inflammatory demyelinating neuropathies. And so in the lab, Jérôme Deveau used a rat model for the Guillain-Barré syndrome and analyzed the distribution of cell adhesion molecule, of nodal cell adhesion molecule in this model. So the Guillain-Barré syndrome includes several neuropathy of axonal origin or demyelinating neuropathy, which are relied with autoantigens, but the autoantigens are not clearly known in the demyelinating neuropathy. So at that time, Jérôme showed that there was, in this rat model, that there was a disruption of the cell adhesion molecules of the node, including neophysin and gliomedin, that precedes during the time course of the disease, which precedes the disappearance of the sodium channels at the node, and which precedes the focal demyelination at the level of the node of Franquier. So the functional consequence is a decrease in the compound action potentials and block of conductions here analyzed in ventral spinal cord. So next, Jérôme collaborated with a Japanese group, a group of Nobuhiro Yuki, and he analyzed a cohort of Guillain-Barré and CIDP patients. So CIDP is a chronic form of demyelinated neuropathy. And so Jérôme showed that around 40% of the sera in Guillain-Barré and in CIDP were immunoreactive against the nodal or paranodal region of delacerated sciatic nerves. So as you can see, this is an example for a Guillain-Barré patient. So AIDP is acute inflammatory demyelinated polyneuropathy. And you can see that the serum of this patient strongly bound the paranodal region. And in fact, using a candidate approach, Jérôme showed that around 26% of the sera were reactive against Contactin or neophysin or enarcane or gliomedin, which are cell adhesion molecules of the node and of the paranode. So here is an example of a serum which is immunoreactive against Contactin. So at the same time, I was contacted by a group, a Spanish group, the group of Isabel Illa in Barcelona, and she determined also that Contactin is an immune target in CIDP. Using a different approach, she used mass spectrometry to identify Contactin as an antigen, which is a target in few CIDP patients. So as you can see here, the immunoglobulins from 3 CIDP patients can bind the paranodal junctions here in the system of myelinating culture of the RGP. So it's just a few patients. It's 3 patients out of 46 CIDP patients. But these patients form a subgroup with a very aggressive form of CIDP and poor response to intravenous injection of immunoglobulins. And moreover, they are IgG4 immunoglobulins. We will see later what it could mean. So we start a collaboration with Isabel, and we first ask what are the molecular determinants which are targeted in these patients. So we use our system of neuroblastoma cell transfected with Contactin, either the immunoglobulin domains or the fibrinogen domains, and we observe that in 3 patients the immunoglobulins were directed against the immunoglobulin domain specifically. Next, cells were treated with tunicamcin to completely prevent N-glycosylation. But it also prevents the export of contactin at the cell surface. So we did this immunofluorescence staining after fixation and permeabilization, and we observed that the immunostaining was completely abolished after treatment with tunicalcin, meaning that the autoantibodies are directed and glycosylated form of contactin and against the immunoglobulin domains. So next we analyzed, we used that the N-glycosylation mutant that we generated, and we observed that in one patient, the immunoreactivity was completely abolished. I don't see I don't know if you see the staining, but specifically on the cells which in which contactin was expressed, this mutant was expressed, there is no more immunostaining. So it seems that these autoantibodies specifically react against this cluster of N-glycans here, and this cluster of N-glycans is precisely implicated in the binding of the glial ligand, the neuropathin 155, meaning that these autoantibodies may block this interaction. In another patient, we determined that the immunoglobulins bound to the complex of contactin and Caspr expressed in immunoblastoma cells. But when contactin was expressed alone, it was not immunoreactive. Next, we tested whether contactin transfected in mutant cells could be immunoreactive. So in this cell, contactin bears mannose-rich N-glycans, and it is immunoreactive for this patient 3. So this means that these autoantibodies specifically react against the high mannose form of contactin. And again, it's interesting because this is the high mannose form of contactin which is implicated in interaction with neuropathy, meaning that these antibodies, these autoantibodies, may block the functions, may block these interactions. So if I summarize this data, the anti-contactin autoantibodies from these 4 CIDP patients are specifically directed against functional modules. We don't know if it's a glycopeptidic epitope or a conformational epitope which is acquired due to N-glycosylation. So next, we ask what are the mechanisms implicated in the pathogenicity. Well, these 4 antibodies are of IgG4 subtype, meaning that they could not induce a complement-mediated attack. So it could be that these antibodies may be function blocking. So we set up an aggregation assay to examine whether these autoantibodies may be function blocking, and we transfected neuroblastoma cells either with contactin and Caspr GFP on one side, or with a neuropathin 155 fused to M-shell in another batch of cells. So the cells were mixed together and incubated for 2 hours in a rotary shaker, and we observed that the cells could form mixed aggregates which are induced by the interaction here, by the adhesive interaction. And we showed that the purified immunoglobulins for the patients were able to block the formation of the mixed aggregates at different concentration depending on the titer of autoantibodies in the patients. So these autoantibodies are function blocking. So next, we analyze whether they may induce alteration of the formation of pyramidal junction, and we use a myelinating cell culture. So it's DRG neurons which are myelinated by Schwann cells. So this is experiments which were performed by Bruno Hilbert in Berlin. And you can see that the myelinated segments are stained using MBP, a myelin marker, which is myelin basic protein. And you can see the numerous nodes and paranodes here, which are stained for Caspr and for sodium channels. So what we observe is that it's really a mature culture after 6 weeks in vitro. And what we observed is that after 1 day of incubation with immunoglobulins for the patient, we observed an alteration of the immunostaining for Caspr. So we did a quantitative analysis and measured the immunofluorescence for Caspr related to immunofluorescence for the sodium channels, and we observed a significant effect. So it seems that these autoantibodies could induce an alteration of paranodal junctions by perturbating the interaction between Caspr, Contactin, and neuropathies. And this was especially interesting because these autoantibodies are of the IgG4 subclass. So we can now really strongly argue that these autoantibodies against the paranodal antigens may really participate in the etiology of CIMP. In the switches. So in the second part of my talk, I will show you some recent data which are still not published and which were obtained using this in vitro system of myelinating DRG culture. We were interested in analyzing the assembly and the stability of the paranodal junctions, and also, as you will see, of the juxtaparanodal region using this system. So here is the phase contrast image of this culture. You can see here the myelinated segments here. This is the representation of the Schwann cells, which is migrate first along the axon and then wrap the axon and make several rounds of myelin around the axon. And at both ends of these myelinated segments, this is the myelin terminal loops which are making the septic junction here. So here is a immunofluorescence staining of such a culture with the myelinated segments which are stained for MAG, which is a myelin marker. And at both ends, you can see in green the paranodal junctions. We call iminodes here because there is only one side of the node, and here, for example, it's a node. We induce the expression of Caspr GFP using an adenoviral system and a normal specific promoter, which is synapsin. And we induce here the expression of Caspr, which is recruited at the paranodal junctions. So it was really important to make a video microscopy and dynamic analysis of the formation of these paranodal junctions. Hope that it will work. This is the movie. So we analyze here by video microscopy the compaction of a myelin loop. So it's a movie during 6 hours, in fact, and as you will see, there will be a compaction of these myelin loops. I will show you the movie. So as you can see, the myelin will be the terminal paranodal loops will be compacted as in as in a spring, but the paranodal loops remains really stable during all this time, and they are in fact very stable structure. So the compaction seems to be generated by the contraction of the Schwann cells here in the middle. So next we did some fluorescence recovery after photobleaching experiments, so FRAP analysis, to measure the lateral stability of Caspr. So I don't know. There are some modification of the slide, but here it's so here it's an unmyelinated axon, and we measure the lateral mobility of Caspr GFP using here photobleaching in a small region of the axon. And as you can see, there is a strong recovery of fluorescence. The mobile fraction is around 73%, meaning that Caspr GFP is highly mobile within these unmyelinated axons. But when Caspr GFP is recruited at the level of a node, of a paranodal junction in a heminode or in a node, at that time the Caspr GFP becomes completely immobile, with a mobile fraction which is only 30% or 9%. So because Caspr is associated with Contactin, and Contactin is a GPI encode molecules, the complex, we show that the complex is recruited within the lipid route. So we test whether RAP disruption may induce some alteration of the stability of the complex, but it is not the case. When the cell culture was treated with methyl beta cyclodextrin, which disrupt RAFT, which is trap cholesterol, as you can see, these paranodal junctions remain completely, the paranodal junction component remained completely immobile at that time also. So next we tested the possible role of protein 4.3 in the stabilization of Caspr at the paranodal junction. So we collaborate with Laurence Goutte-Rose in the group of Jean-Antoine Giraud. So Laurence examined the phenotype of knockout mice, conditional knockout mice for 4.1b, which is deleted specifically in the neurons. And protein 4.1b is a scaffolding molecule of FERM family, the family of 4.1 ezrin, radixin, moisin. And these proteins are indicated, are associated with Caspr and with Caspr2, both at the paranodes and juxtaparanode. And as you can see here, this is an electron microscopy image of the paranodes. You can see that the transverse bands are present in the knockout mice, and that there is mostly alteration of the juxtaparanodal clustering of the potassium channel, meaning that the paranodal junctions are quite are not disturbed in these knockout mice, but the aggregation of potassium channels at juxtaparanode is strongly altered. So we tested the knockout mice for 4.1b for the mobility of Caspr at paranodes. And so this is the immunostaining here for 4.1b in the myelinated axon. You can see that it is strongly recruited at the level of the paranodal junctions. In unmyelinated axons, well, the Caspr GFP is strongly mobile, as in wild-type animals, and also as in wild-type animals, Caspr recruited at the paranodal junctions remains really stable. The mobile fraction is around 11% in the knockout mice. So meaning that 4.1b does not play a strong role in the stabilization of Caspr in this region. So maybe it is because of redundancy between the protein of 4.1 family, because 4.1g is also enriched at the paranodes and may compensate for the lack of 4.1. So now I will show you some data about the formation of juxtaparanodal complex in this cell culture. Well, the juxtaparanode have been well studied for example, in the sciatic nerve or in myelinated axon in the central nervous system, but it's difficult to obtain, it's very difficult to obtain the clustering of juxtaparanode in a cell culture system. So this is the work of Bruno Hiver, who set up a system, we set up a culture conditions in which we can observe the clustering of potassium channels in myelinated fibers Here you can see that after 4 weeks in vitro, we observe the concentration, the clustering of potassium channels at the level of paranodes, and also here at the levels of the juxtaparanodes and paranodes. This is a double staining in green for the potassium channel, and in red this is Caspr as a paranodal marker. Next, in more mature culture, you can observe here that the potassium channels are nicely clustered at the level of the juxtaparanodes. So they are progressively excluded from the paranodes and restricted to juxtaparanodes, as was also observed during development, in fact. So what we observe here is the colocalization of the potassium channels together with Caspr2, which is also clustered at juxtaparanode here, and with 4.1b, which is both located at the level of a paranode and juxtaparanode. But despite our efforts, it was not possible to obtain an immunofluorescence staining for TAG1 in the glial partner of Caspr2 axonal and glial partner of Caspr2 at juxtaparanode. But we will still try. I hope that it will be possible to get it. So here is, I don't know, if the red channel is not really easy to see. Here it's a double staining for 4.1b and KV1, just to show you that 4.1b is really located at both at the paranodal and juxtaparanodal side. So it's not easy to understand why the potassium channels is restricted at the juxtaparanodal junctions. We don't know exactly what are the scaffolding protein which may be implicated in this regional distribution of the potassium channels. So here's just an image to show you that the juxtaparanodal component, the potassium channels, are excluded from the paranodal loops progressively. So you can see in green the potassium channels, which are in between the paranodal loops, and then it is excluded from the paranodal region, and it is juxtaparanodal. And finally, what we observed is that in some nodes, we can see a stronger staining of the potassium channels at the nodal site. So it's in culture and cell culture after 4 weeks in vitro, and you can see here the nodal staining for potassium channels, which is progressively eliminated from the nodal region. So this is the schematic drawing of two myelinated segments here. Yes, I get it. I get it. No, I lose it. You see something? It is very small. So this is a schematic drawing with two heminodes of adjacent myelinated segments, which are becoming contiguous. Here, the myelinated segments are contiguous in this picture, and here there's an unmyelinated segment between two heminodes. So what we observe is that the potassium channel are frequently trapped between two paranodes at a nodal position. This is also the case here for Caspr2, which is also trapped at the nodal position, meaning that Caspr2 and KV1.2 are first addressed to the cell membrane, can diffuse freely all along the axon, and are trapped between two paranodal junctions. So they are trapped even at the level of the node or at the level of the internode at the internode. In contrast, the scaffolding protein 4.1b is always excluded from the nodal region. Here is the double staining for KV1 and 4.1b. And as you can see, the scaffolding molecule 4.1b is not located at the nodal region. It is quite clear from the unmyelinated segments and only concentrated in the myelinated segments, maybe interacting with nectin like a protein which implicated in interaction between the myelin and the axons. So it seems that it is not a preformed complex that is addressed to the cell surface, but progressively Caspr2 and the KB1 and TAG1 may interact with the scaffolding molecule for contamination. So it's not clear at the present time how this juxtapiramidine is formed, but we hope that we have obtained some insight into the formation of this complex, and we hope that we could continue to collaborate with Donna to understand more and more how it is formed during myelination. So I would like now to thank the people of my group. So, well, Bruno is in the room. Is he on the picture? I'm not sure. And well, I would like also to thank Jérôme Deveau on the left, who is a specialist of a potassium channel and of neuropathy, of demyelinated neuropathy. I would like to thank a PhD student, Delphine Pinatel, and the previous PhD student, master student, Karine Bonan and Christophe Bell, also Marine Lavas, postdoc in the lab. And for the fly, for the Drosophila story, I would like to thank Monique Laval. And also my collaborators, Laurence Boutbrose and Jean-Antoine Giraud in Paris, Mansour Bat in the States, and Isabelle Ilia in Barcelona. So many thanks for your attention.
Questions?
So can you, in this last part where you follow the segregation of the potassium channels, is there a way where you can express maybe on top of it, let's say, some of your mutant forms of Contactin or Caspr or something, and see different?
Well, it's difficult to, it's a system which is not easy to work with. It's difficult to express recombinant protein in this myelinating system, so we use adenoviral protein. So we tested with Caspr GFP, it works very nicely. But we try with Contactin GFP and Caspr2 GFP and Larkin GFP, and it was not so good. So with Contactin, the problem, and it's the same thing with Taiwan, is that it's a GPI molecule, and it's difficult to put a tag, an epitope tagging which does not perturb the interaction. So that's a problem with Contactin. Put GFP in the extracellular domain, and it perturbs, I don't know how, but it's not recruited at the final action. So it's a difficult system to work with. Yes.
This trapping of potassium channels in the nose sometimes, is it a particular type of potassium channel? You see it with 1.2 only, or do you see it in vivo ever, or has anybody seen it?
Yes, it is the complex of 4.1b, it's the complex of KB1, 2.1, 1.2,
okay.
exclusively. And it has been also described in mice. This is the work of Vatnik and the work of Schrager. And he also observed during the development at B5, for example, many nodes, I forget to mention this, he observed the clustering of the potassium channel, transient clustering of the potassium at the nodal region. And it could play, it could make sense to prevent hyperexcitability of the nerve during development.
Okay.
So it could be.
But they are cleared there after the sodium channels, or?
Yes. Yes. They are co-expressed in
More questions? No? If not, thank you very much.